Thank you, Chris. Good afternoon, everybody. And because this afternoon, I think the last section, I think is the innovation in diagnosis. So in my talk, I just would like to share a piece of ongoing work in my laboratories about how can we use matured high technology or new technology to resolve some of the technical issue inherited by some traditional assay, which we use quite daily in our diagnostic lab. So in my talk today, I just to show you how can we use real-time PCR to help us to resolve some problem when we do the VNT test in our routine diagnosis or vaccine match. So in my talk, it's quite simple, and it's into the three parts. So I'll give you a little background why we do this, and then can share some results with you, and then simple conclusion and some of the future work we are going to do next. And probably we heard quite a lot yesterday and today about the virus neutralization test. And I think this test, of course, is used for detection of EuFMD neutralization antibodies for the measurement of the serological response to the EuFMD infection, and also for vaccination, particularly if you want to monitor the immunity in vaccine animals. Probably the VNT is most recommended method to do this work. And of course, I just mentioned this VNT is also quite widely used for the EuFMD vaccine matching test in vitro. So we heard quite a lot about this last 2 days. And particularly also in the EuFMD-free countries, probably this VNT also can use for confirmation of clinical diagnosis, and also can provide some of the information about epidemiology in the field. So so this test we can consider is a confirmatory test. Particularly when you do the ELISA test, sometimes if you got some negative results and you're not sure whether it's positive or negative, or true positive or false positive, normally go for the virus neutralization test. And also you do the 3ABC test. Normally, if you got some positive, you need another assay to confirm your results. So the virus neutralization test is the most recommended assay that you can choose. It's very highly specific, so it's a very reliable test. But probably everybody, if you do the VNT test in the lab, probably agree with me is this test is quite time-consuming, and you take quite slow. It takes about at least 2 or 3 days before you can get the results. And another one is you have to read CPE on the plate, because you look at the virus infectivity on the cells. So you have to read on the microscope, which is also another headache if you got quite a lot of samples, and if you're not familiar with the CPE induced by EuFMD, you're quite nervous. So which one's CPE, which one's not. So it's quite easily infected by the man-made effect. So sometimes different laboratories, you can compare the results. Sometimes it's not the same. It's quite variation is quite high from lab to the lab. So how can we sort out this problem? And because one technique we're thinking of is real-time QRT-PCR, because nowadays the real-time PCR has been widely used in our veterinary diagnostic lab, particularly in EuFMD. If you cannot isolate the virus, you never go for the real-time PCR to detect the virus genome, which can give you some indication whether the sample is positive or contain the virus or not. And this, again, because it's very sensitivity is very high, also very specific for the EuFMD detection. So now it's widely used. And the result you can get within hours if you particularly if you use the automated RNA extraction, even much faster. Importantly, I think another features of these assays, you can quantify the virus in the samples, so which give you some information quantitatively how much virus in your samples. So, because this good things for these assays, so we want to use this technology to help us to resolve some of the technique headache inherited by the VNT test. So that's why we tried to use the RT-PCR to quantify the reduction in the virus replication resulting from the neutralization antibodies in the serial samples. So that's our aim, whether we can use this assay to help us to see the positive sample or negative sample. So it's quite simple test. Apart everybody knows, first two steps is more or less like the traditional VNT, but we terminate VNT test at 20 hours after inoculation. So I'm going to talk a little about later on why we used 20 hours after infection. And after that, we just get RNA extracted from the cells, and then we do the quantification of viral RNA in the samples. Of course, to see the difference in terms of reduction of the virus replication in the sample compared to the absence of the sort of the virus control, we need to calculate do some calculations. So what we do is we use percentage of the reductions in the EuFMD RNA copy numbers in comparison to the copy numbers in absence of the samples, which is virus as control and no virus neutralization activities in those samples. And then we can get the percentage of the reduction of EuFMD RNA copies in the samples. And then we can decide positive or negative. So in that case, we have to decide. To the first step, we have to talking about the results, what we have so far. So first, we have to look at whether this RT-PCR its performance is good enough for us with our local virus, which we isolated in China. So the real-time PCR, RT-PCR we use is based on the 3D, so which means we use the primers targeted EuFMD 3D genome, which I think this is one of the widely used assay globally, because another one is iris primers. So the 3D is one of the two most popular RT-PCR assay for EuFMD RNA detections. So on the right-hand side, I think, from you. And we just wanted to see with the standard 3D RNA to validate the performance of this real-time PCR. From here, you can see the range of the virus standard RNA expanded from 6 logs, ranging from the 2 to 8 logs per reactions. So which is quite similar. Previous report is quite wide range of the detection. So which is good for us because we want the wider range of detection. Otherwise, it's not very good in terms of sensitivities. So after this, we repeated this a few times, and you can see the variation is not very big, and it's within a very good performance. After that, we move to the further validate this performance of this assay, real-time PCR assay. We use the local virus and extract the RNA from the virus and make the 10-fold dilution from 10 to the 1 to 10 to the 7, and then performs the real-time PCR. And again, you can see that we are performs each time. Each dilution, we use 3 different tests, and then we repeat a few times on the different day to make sure the reproducibility is good enough for us to do the further validation. So again, from here you can see the test is good. So we're happy with the performance of this test. And of course, because we want to use this QRT-PCR to look at the virus replication. So the next step is we'll have to look whether we can, when we're going to stop the neutralization. And we have to make sure the time, because the purpose of the work is want to speed up the VNT test. So we want to look at within the 24 hours, which time point we should stop the test, and then they can do the virus detections. And from here you can see when the 1 hour after infection, we use the IBR2 cells and inoculate cells with 100 T-side50, which is it's similar to when we do the VNT test. We use 100 T-side50s. And then we looked virus replication and the different time of infection, and we look at in terms of the virus copy numbers. And the day 1 hour after infection, and roughly you can get the 10 to the 3 copy numbers, and gradually and increase to 10 to the 7 copy numbers, and 20 hours after infections. After that, we didn't see too much increase in terms of the virus copy numbers. So that's why we decided we use 20 hours after infection as our ending point to analyze the virus neutralization test. And 20 hours, I think it's good for us because we can do it test just 5 o'clock, and then we can read the result next morning. So it's convenient. So we're happy with the performance of the real-time PCR on the virus RNA detection. So now next step, we're applying this assay to the VNT to see whether we can use this assay to quantify the reduction of the virus replication resulting from the neutralizing antibody in the serum. So we do this different test, a few tests to see whether it's good enough for us to do the further work. So first of all, we just look at the negative serum samples. So we do the virus incubation with the serum sample, and to see which dilution of the serum we should use, because probably most people know when you do the serum test, you have to make sure you have to avoid non-specific activities or bindings. Just the ELISA test, when you load the dilution of the serum, you always see some non-specific impact on your test. And for our assay, again, same. Sorry. And when there's a dilution of the serum, and there's just virus load copy numbers. There's a sheep's negative sample from sheep. There's part from the cow, and from pigs. But in China, sometimes it's quite difficult to get this negative serum because in China we use massive vaccination campaign. So for us, sometimes it's very difficult to get negative samples. So we'll make sure the negative, we have used the 3BC screening test to make sure there are no anti-3BC antibody in the serum, and also use liquid phase blocking ELISA to make sure they are negative. And in some cases, we'll have to use the VNT test to make sure the sample is negative. And from here, you can see they are all negative by VNT test, traditional VNT test. Here you can see from the 1:4 dilution up to the 128. There's a control is without serum samples, so with the virus control. The copy numbers of virus here is roughly about 8.3 logs. And from here, the order more or less the same. When the serum diluted at 132, and the virus load more or less quite close to the controls. And this the all the samples. When they do the average, again you can see there's it's quite a variation. It's not very high, and also under the 132, the more or less the virus copy numbers in this particular dilutions is quite close to the virus controls. So in that case, indicate when without these assays, we have to use 1/30 dilution to carry out the further test. And if we do the calculation of the percentage of reductions in the virus copy numbers in the samples over here, you can see the percentage of the reduction of EuFMD virus copy number. It's dependent on the dilution of the serial sample. So the more dilution you did, and you can see the percentage of reduction is getting less. Here you can see that just mentioned the 1/32 dilution, and the reduction percentage just 8%, but they're also quite close. Even the 1/46, 1/128, the model is the same, and they just another converted into the column. You can see here after 1/32 dilutions, and the model is the same. So we are decided to use this dilution to do the further validation, but we need a cutoff for this assay. So what we do is, it's because this just ongoing work. So initially we just the cutoff value was set by N3 SD, which means in this case, because this virus we use the H1, and we cutoff values we consider is 23% in reduction. If it equal or less than this reduction rate, we consider as negative samples. And if it greater that, this value we consider as positive. So with this mind, we do the further test with positive samples, which is we infected from the peaks, infected experimentally with the EuFMD H1, and we collected serial samples and 35 days after infection. And here you can see, again, there's different dilution of serial, and of course, if you look at 1/30 dilution, and the reduction rate is still greater than 60%, so which means it's still positive. And here you can just see that for the positive sample, because they are all quite strong positive by the VNT, and you can hear that even 1/32 direction is still quite you can see that the virus reductions due to the neutralization activities from zero still quite strong. So they are all positives, and we do the further validation with positive serial samples with 4 different positive samples, do the series dilution, and to see the sensitivity, analytical sensitivities in this test. So again, with harvest cells and 20 hours after infection, and extract RNA and the real-time PCR. From here you can see the sensitivity, but they're all positive by the VNT, and you can see that's different, and this end, which is matched with the VNT. If you got a high titer in the VNT, and then you can dilute it further. But in a sense, you can see it's got a wide range of the test, so which means the sensitivity is acceptable. It's good enough for us to do the further test, because from 1/16 to quite a wide range, which means the sensitivity is it's good. And so if we do the more test with the positive VNT positive sample, which also again from the pigs and the cattle infected experimentally with the H1 local isolates. And here just gives some of them and just illustrate the performance of this test. And here you can see that all the VNT positive, and this is just the virus copy numbers in each samples per reactions. And when we do the calculation percentage of the reductions in RNA, FMD, RNA copy numbers, you can see here they're all greater than 23% in reductions. They're all, in a sense, are strong positives. And yep, almost finished. And so that's just another serial sample from sheep. And one is positive, it's not positive, inconclusive by VNT, and 3 of the negative by VNT. Here you can see this one is in our assay is positive, and others also negative. So it's quite more or less the correlation with the VNT is good. And of course, you always have talking about how this, whether it's specific for the Asian one. So we use the Asian one virus with the incubated with the A type seropositive and O type positive. So you can see here, even the all indicators are negative because the virus load is quite close to the control, and if you do the percentage of reduction, they're quite close to the negative sample, less than 23%. And we did a little bit of correlation in terms of virus titer, VNT titer, and our ACE titers. From here, you can see the both tests are quite close to each other. But well, just very preliminary data, so we're still working on with more samples. And there's a conclusion. So I think one message you can take with this combination, traditional VNT with the real-time PCR, you can get VNT neutralization test within a day. So which I think is an advantage, particularly you're talking about FMD, you can heard many, they spread very faster, so you can get the result very quicker. And what we'll do is we'll just now validate assay with more samples, and also we'll do the serotype O and A. So hopefully next time we can have more data to present to everybody. And finally, I would like to thank my team, particularly my student, Mr. Song, and who did the most job. And also we got a fund from the National Research Fund and also Innovation Fund, a master's degree studentship. Thank you for your listening.
Thank you very much, Zhang, for presenting this interesting combination. Are there any questions? Yes.
Nope. Yeah, that's fine. Yeah.
So yeah, thanks for the presentation. It's an interesting approach, but I suppose to be devil's advocate would be to ask two questions of this new sort of process. Is
yep.
Presumably the cost is much more, so you're now using molecular agents alongside serological tests, which
yep.
are conventionally quite cheap. So that seems to be a bit of a disadvantage. But also, I think most of us who've tried to put serological tests alongside molecular tests will recognize that a lot of serological assays have a lot of virus floating around in them, and it's just,
yep.
Do you also see problems with cross-contamination with this test?
And because I think in terms of if you do the virus RNA detection, yeah, we do have this problem as well in the lab, and that's why we'll have to make sure they follow the sort of proper procedure to make sure there's no cross-contamination. And in our hands, because we got enough space, so at the moment we don't see there's a big problem for us in the lab. And in terms of the costs, of course, agreed, yeah, but the PCR now is not very expensive, but it's like additional equipment. Reagents are not very expensive, but it's much quicker. So in that case, if you have outbreak or you want to do something quicker, so you have to balance the cost for benefits. So it's dependent on the situation. You agree that it can be more expensive if you combine both.
Okay. Any other question? If not, then I thank you again, Zhang.
Thank you, Chris.
And I hand over.